I'm Andreas Schmidt, and this is my colleague Jonathan Scolnick. We are from Singleron Biotechnologies. It has been amazing to be here at ASH and look at a lot of new discoveries on multiple malignancies and diseases. We, with Singleron Biotechnologies, focus particularly on single cell analysis, combining RNA as well as protein on a single cell level. And what makes us special here as a company is that we provide everything from devices and reagents all the way to like very complex bioinformatic analysis. And that's typically the crucial point of it, since these are huge data sets that then have to be reduced to clinical findings that are really meaningful. This year we presented here 3 different stories unfolding single cell for particularly multiple myeloma, which is a very heterogeneous disease where single cell technologies are of particular value. And my colleague Jonathan Scolnick will share with you one example that we earlier presented today.
So thanks, Andreas. In the poster I presented, we were collaborating with Professor Chung-Wee Ju's group at the National University Hospital in Singapore. And the goal was to use single-cell multi-omics to try to identify factors that in the immune microenvironment that correlate with progression-free survival, specifically in translocation 414 myeloma patients. Now, when we started the project, we sort of immediately identified that with all of the different samples that had been come from a cohort that had been collected over multiple years, there were huge batch correction problems. And yet the existing batch correction methodologies that we had tried were all overcorrecting the data. So we actually had to go back and develop an entirely new batch correction method that was more conservative than the existing methods. And once we did that, actually, we were able to look at the data and clearly identify a very small population of T cells that correlated with progression-free survival in these patients. And these T cells actually represented less than 1% of the total population of cells that we collected, but there was a very clear difference in the number of T cells between patients who performed well under therapy and patients who did not. We then went on to test these T-cell prognostic in a non-414 cohort of patients and found that it maintained its prognostic value. And so now we're moving on to testing that in a much larger cohort.